After about 24 to 48 hours, your plates, if we've done this correctly, will look something like this, where you see single colonies on your plate. And so the goal of this lab is to identify what those single colonies are. And in order to do that, we need to first extract the DNA of a single colony and then perform a polymerase chain reaction on the 16S ribosomal gene and send it off for Sanger sequencing. So that's what we're gonna do.
Okay, can you explain polymerase chain reaction? Yeah, let's just start there.
Okay, so PCR, polymerase chain reaction, it's a very common lab technique. It's basically used to amplify DNA. That's all you're doing.
And by amplify, you basically just mean making lots of copies.
Yeah, like millions. Yeah, making millions of copies of a single gene. And the reason why that's useful is that when you send it out for sequencing to identify what the DNA code is of that gene, you want lots of copies because that technology requires it. With PCR, you can use certain primers, which are small DNA sequences that target the gene that you're trying to amplify. So in this case, we're interested in amplifying the 16S ribosomal gene in bacteria because that is a very commonly used gene to distinguish one bacteria from another. A different gene is used, for example, with yeast, so you wouldn't use the same exact gene.
So essentially what you're doing is we originally isolated cells from the kombucha sample.
Yeah.
Now we have here colonies that have formed from those cells.
Yeah.
And now what we're doing is we're taking DNA of those cells and then further taking just a small segment of that DNA.
Yes.
And in particular, we're targeting a segment that will help us be able to identify what bacteria, what specific species.
Every bacteria has this gene. It's a ribosomal gene. It's a gene that is used to encode segments that make the ribosome. And so every bacteria has the 16S gene, but they have different versions of it. And so based on what version of the gene that they have, which we'll find out based on sequencing the gene, we can map that version of the gene back to the bacteria that it came from.
And you're essentially comparing this with like large data banks that already know what gene is associated with what species.
Exactly. So first, in our tube, we have some Prepman reagent, and basically this has all of the buffers and chemicals that are required to break down the cells, because when we want to extract the DNA from the bacteria, we want the bacteria to essentially burst and give us all of their DNA. And so this is a mixture of everything that we'll need to get those bacteria to burst. You can go in and pick one single colony. For example, I'll pick this one. So that's the first thing. You've taken a single colony and you put it in here. The next step is to vortex it and then put it in the heat block. So this is a vortexer, also another very commonly used instrument in the lab, and its only purpose is to help you like mix tubes up and kind of homogenize a mixture. So in this case, we want the bacteria to homogenize with the liquid that we have it in. You can think of this as like a mini blender. So when,
like almost the opposite of the centrifuge.
Exactly. Yeah, so you can think of those as opposites. The centrifuge separates, and this kind of brings things together. So you can use this for anytime you have, two liquids or like a, tissue sample that you're trying to mix with a liquid, whatever it is, this is a very useful tool. All right, so the next step, once we have our mixture homogenized with the reagent, we need to boil it. And the way that we do that is through putting our tube in this heat block. And a heat block is essentially just metal that heats up to whatever temperature you would want it to be. So in this case, since we want to really burst the cells, we're going to use heat to do that. And this heat block is set to 100 degrees Celsius. So that's basically the temperature at which water boils. And we'll leave this for about a minute until the cells disrupt.
So the combination of these chemicals and the heat is just trying to burst the cells.
Mm-hmm. Yeah. And after we burst the cells here, we're gonna go back to our centrifuge and spin it. So now we're gonna get a pellet again, right? Because the centrifuge makes pellets. We'll make a pellet at the bottom, which will be all of the cell debris, like the cell gunk that we don't want. And everything that we do want, which will be DNA, will rise to the top. So we're gonna actually keep the liquid this time and discard the pellet. So now we've come back from the centrifuge with our DNA, which will be in the top layer, the supernatant. And all we have to do now is take a small sampling of our DNA and add it to our PCR tube. So the PCR tube and this bead already has all of the ingredients that we'll need to run this PCR reaction, other than the primers and the DNA. So we need to add the DNA to make sure that this actually works. So the pellet contains Taq polymerase, which is the polymerase that is required to make the copies of the DNA. It has some salts and some buffers, and then it also has GNTPs, which are the nucleotides that get incorporated into the DNA by the Taq polymerase. Yep.
And those together are like the magic to making millions of copies.
Exactly. That makes like the complete recipe for doing a PCR.
And it's only making millions of copies of that specific gene that you're looking for.
Yeah. So like the purpose of having those 16S primers is that small section of DNA is what actually gets copied.
But the rest of the DNA is in there. It's just
still
not
in there.
Getting copied.
Exactly. Once you're ready to run your PCR reaction, you go to this machine, which is the thermocycler. So a really important thing about this thermocycler is that it does that, it cycles. So as we can see here, to run a PCR reaction, there's really 3 super important steps, and they all occur at different temperatures. So the first one is this at 95 degrees, and this is the denaturation step. So basically it separates the DNA strands from one another. And next is the annealing step at 60 degrees. And so that's when the primers are able to anneal to the DNA. And then finally, the 72 degree step is the elongation step. So that's where the primer is attached, the DNA polymerase finds where it needs to go, and then the DNA is able to get copied. And in this case, it's the 16S gene that we're making hundreds and hundreds, actually thousands, maybe even millions of copies of these that we can then use to further study the bacteria. So the thermocycler opens up like this, and you can add your little tube right in there. Now that our PCR reaction is complete and it has been running for about 2 hours, we need to do a quick cleanup step to separate out the amplicon, which is the 16S gene that we've been amplifying, from the rest of the materials that were in there. So to remove the Taq polymerase, to remove any extra salts, to remove the DNTPs, and make sure that when we send this sample out for Sanger sequencing, that it is as pure as possible.
What do you mean by sequencing?
There's a lot of different ways to decode the DNA of specific genes. It's really useful for sequencing small chunks of DNA, like the 16S gene. And then if you want to sequence whole genomes, you're basically doing Sanger sequencing in parallel hundreds of thousands of times, and with slightly different setups, but the idea is generally the same. It's all, each nucleotide emits a certain wavelength, and it's picked up by lasers and fancy equipment, and that happens massively parallel when you're trying to decode an entire genome, because there's billions of base pairs.
And so essentially your end product will be like A, C, T, G, and the sequence that they're in.
Yeah. Mm-hmm.
I'm having so much appreciation for the people that like figured out this method. Like,
yeah, it's amazing.
Pretty unreal.
Yeah.
When you actually think about how many steps there are and how many possibilities there are to mess up if you're like actually trying to come up with like what you should do.
Right. Yes, definitely.
Oh my gosh. Okay.
After we get our samples back from Sanger sequencing, we're going to blast our 16S sequences into the database and see what kind of bacteria we can identify.
What do you mean by blast?
Okay, BLAST is actually an acronym, and it is the name of a specific tool that you can use to match DNA sequences back to the animal, the bacterium, the yeast, the whatever creature it came from. So when scientists do any kind of whole genome sequencing or even smaller Sanger sequencing, these sequences are uploaded to a database. And so when you blast something, for example, our samples, you're trying to match your sample to one that has already been deposited in the database. And the important thing is that when it's deposited, it's given, you know exactly what sort of species and subspecies and specific organism that sequence came from. So it can help you identify where this DNA came from.
This is like a ever evolving and growing database. Thanks for joining us today. We hope you learned a lot from these videos, and let us know if you have any questions.
We had a blast.